Hello! So I think that we are live and streaming. Welcome to the second part of this two-part news video experiment. If you missed the first video on Tuesday, the idea is that on a Tuesday I will summarize a recent-ish genetics news story, and then on a Friday I will do a live stream to answer your questions about it. So if this is working, if you can see and hear me, say hello, say hi in the chat. My whole idea with this is that the most fun thing for me is to actually talk with people about science rather than just talking at you about science. So I'm hopeful that this is a great way to be able to do that. Okay, great. We got a hello in the chat, so I think that this is working, which is awesome. A couple points of business first. This week, we're going to be talking about the LilBUBome. So this was the genome sequencing of one of the internet's favorite cats, Lil Bub. But hopefully, I think this is like a really cool story to start with. I think it's really approachable and a really great jumping off point to talk about genome sequencing and mutations and a whole bunch of cool stuff in genetics. But in the future, I want to jump into a lot of different types of genetics and biology stories. So if you come across a story that you want to be featured in something like this, please let me know. Ideally, I think that this is like a perfect 20 to 30 minute kind of format, but you guys sent in so many awesome questions that I want to see how many we get through and sort of how long this goes. Hello. Oh, there's so many wonderful people here that I recognize. Hello. I'm so excited. I have two different screens running here, so you might sort of see me looking around, and I've got notes and the annotated paper and all sorts of good stuff. A couple other orders of business before we dive in and get started. Number one, this is just sort of a cool tidbit and fact. I actually started my YouTube channel on February 14th of 2007, so in a crazy coincidence, this is in fact my 13th anniversary on YouTube, which seems kind of cool. And two, this is only my second time ever live streaming on my YouTube channel. So we're going to figure this out together and see how this goes. But the thing that gives me confidence that this is going to work is that you guys are such a wonderful community that I think you're going to be in this together. So I'm really excited. So let's jump into the actual science here. I want to sort of give you a quick roundup summary of the paper before we dive in. So if you did miss the video on Tuesday, Lil Bub is an internet cat that you might recognize. I'm actually going to put one of the figures from the paper up on the screen now. These are some pictures of Lil Bub. And if you notice, she has some unique characteristics about her. So she is sort of a dwarf kitten. She's much smaller than normal cats, and she sort of retained this kitten size into her older life. She has some bone phenotypes, so she has osteopetrosis, so she has these sort of built up thicker bones, and she also has polydactyly, which means that she has multiple additional digits on her paws. So scientists are really interested to figure out, is Lil Bub, does she have these different changes and phenotypes because of just one mutation in her genome that is giving her these multiple phenotypes, or is it multiple mutations that are giving her these multiple phenotypes? So a group of scientists decided to sequence her genome, and they found that in fact there were two separate mutations that they think are causing both her osteopetrosis and her polydactyly phenotypes. So this is pretty cool, but one of the things that really attracted me to this project and to talk about it is the fact that this was not only done for scientific reasons, but also because the authors wanted to use this as a great science communication project to talk about how we find mutations in rare disease and what genome sequencing is, and to try and have this project have sort of this other life as a science communication project, which to me was really, really exciting. So one of my favorite parts of my job is the fact that I actually get to talk with scientists about the work that they do. So I reached out to the scientists who were involved with this paper, first of all, just to ask if I could use their figures and talk about this. And they were kind enough to answer some of the questions that you sent in. So some of the questions that you sent in, they've actually already made videos about. So I've put those in the description to this video so you can go and watch them afterwards. And I'm going to get to some of them too. But one of the things that I wanted to talk about first, I'm going to pull up an email from Dr. Simmons, who is involved in this paper. And she first of all had a clarification to something that I mentioned in the first part one of this video series. So she said, I just wanted to clarify that we actually think Bub's small stature is likely linked to the osteopetrosis. So since the bones calcified more, they probably couldn't grow so much. So the dwarfism may not be a separate trait. But as many of the questions said in some form or another, this is only an educated guess. We can't know 100% for sure. So I wanted to start off with that. Dr. Simmons also sent a bunch of videos that, again, I've linked down below that you can dive into, talking about sort of what their data looked like, how they found these mutations, what these mutations are. But the question that I wanted to get to first, before we really dive into the science here, is that somebody had sent in a question asking about sort of the science communication angle of this. And again, this is one of my favorite angles of this topic. So talking about how the question was, could you go into more specifics about how the project worked and the science communication was shaped and what the reactions to the project were? So her answer to that was that we used a bunch of different approaches. We tweeted, used Facebook and a blog to write and vlog some about the project. We also used the crowdfunding site for blogging as a kind of virtual lab notebook, which I personally think is very cool. At one point, we had a Reddit AMA with a bunch of awesome questions. And during the course of the project, we also gave a bunch of interviews and tried to link to some of the articles in our social media, because sometimes professional storytellers, aka journalists, are just better at summarizing a story than a bunch of scientists trying to do this. So when the paper came out, there was also a pre-light feature on it. This is a platform where scientists read the paper and ask questions, and they also responded to those. And overall, I would say we try to explain as much as we can and also try to make ourselves available to questions, which she so kindly did to my questions. Answering questions is particularly important because sometimes we're so involved in the subject matter that we may not realize the things people want to know. We also love the fact that LilBUBome is sort of taking on a life of its own thanks to projects like yours, which I appreciate. And also, for example, someone used the LilBUBome for some questions on a genetics university course. How cool is that? So I think that that is really, really cool. And I really appreciate that Dr. Simmons took the time to actually reach out and answer my questions and get back to me, which I super appreciated. So, oh my goodness, so many people are jumping in on the chat. I love this. Thank you. Yes, I did get a haircut. But all right, so I want to get into answering some of the questions that you sent in on the first video. So I've sort of tried to group these into sections because there were a lot of very similar questions. So before we get into talking about the specific mutations that Lil Bub had in her genome, there were a lot of questions that just asked, what is a mutation? And a lot of questions that were sort of asking about the nature of mutations and what does this really mean when there are mutations in a genome. So these are sort of some questions that were sent in, and I know I'm going to pronounce some people's names wrong, so I really apologize in advance. But these are some questions sent in by Johan, Neve, Yochen, and Aaron. So what is a mutation? So a genetic mutation, really simply, is just a place in someone or a cat's genome that is different from what most people have in their genome. So when you think about a genome, remember that it's a long string of molecules that we denote A, T, C, and G. And the order of these different base pairs actually encodes for the different proteins that go off and do work in our body, as well as a bunch of other things. So in the human genome, we have 3.2 billion of these base pairs. And the cat genome I looked up a little earlier is, I believe, 2.7. And we have 2 copies of every genome, one from our mom and one from our dad. So this makes us diploid. So we have 2 copies in us of this 3.2 billion base pair genome, and in cats of their 2.7 billion base pair genome. So mutation is really just a spot in Lil Bub's genome where her genome is different from most other cat genomes. So you can imagine that in some places this might be a single base pair change. So maybe an A somewhere in her genome was changed to a T, a C, or a G. So there are mutations like these. These are called, sort of single base pair changes, or single nucleotide polymorphisms, depending on exactly what you're talking about. But there are many different types of these mutations. So let's say you have a mutation somewhere in your genome that changes an A to a T. And let's say that this mutation is in a gene that would actually go on to code for a protein that would do some kind of job in your body. it's possible that this mutation would not actually have any effect on the final protein product. So maybe you have this little mutation, but it's not deleterious. Maybe it's a synonymous change and the protein goes on happily and this mutation has no effect. I think that when we say mutation sort of in a typical sort of social context, we think of it as having a negative connotation, but mutations aren't always bad. And I think I came out of a sort of human genetics lab that was really sort of clinical focused, and we often really try to use the word variant or variation because sometimes that's all it is. It's just a small variant or variation in the genome. So sometimes they have no effect. Sometimes you can have what's called a missense mutation in a gene. So maybe this is going to change one of those amino acids in the protein to something else. Now, again, this might have a small effect on the protein going off and doing its job, or it could have a really big effect on that protein. Maybe it completely changes the structure of the protein and now the protein can't do its job anymore. You can also have things called truncating mutations or nonsense mutations. So these are mutations that might actually truncate that protein. They might put a premature stop signal so that as your body is going along that DNA, turns the DNA into RNA, then the RNA into protein, through the process of transcription and then translation. Maybe it puts in a little signal there that says, "Oop, stop making the protein here," and now instead of that full-length protein, you have a truncated protein. And again, maybe this has a big effect in your body, maybe it has a little effect in your body. There are also kinds of mutations called insertions and deletions. Maybe you have this, long string of bases and a couple are added in, or a couple are taken out. And again, these, depending on where they go in and what they do, can have really big effects or small effects. You can also have big structural variants. So maybe you have a really big chunk of a chromosome that's popped onto another chromosome, or you have really big insertions or deletions. You can have frame shift mutations where, your DNA is read in sort of 3 base pair chunks, and maybe you add in something that shifts over that reading frame. So if you imagine a sentence like, "The man and the cat are fat," if you add in which is all 3-letter words, if you add in a base there somewhere, you might have something that's now "the man and or." If you add in a letter into that sentence, you're going to mess up how you're reading those 3-letter words. So these aren't even like all of the mutations that you can have, but I'm just trying to show you that there are a lot of different kinds of mutations that you can have in a genome. Some of them have really big effects on your body. Some of them have very small effects. Some of them have no effects. It all really depends on where they go in, if they're happening in a gene, if they're happening in a regulatory region of your genome. But really all a mutation is just a change from sort of what we would consider the normal sequence in that DNA. And one of the ways that we figure out sort of what is that normal sequence is by using something called a reference genome. So a reference genome, again, is really sort of looking at one standard genome from a human or a cat that I'm talking so fast. I know, I'm so sorry. I'm nervous to be live streaming. I don't live stream much, so I'm talking extra fast. But sort of this reference genome is something that we have as a standard that we can all compare our data to. Now, this doesn't mean that this is like the healthiest, most perfect genome that has ever existed for a human or a cat. So for example, the human reference genome is, I believe, from 5 people who answered a newspaper article from Buffalo, New York back in the day. And so it's sort of an amalgamation of all these genomes, and we know for a fact that that reference genome for humans actually contains some places that are potentially sort of disease associated. And I don't want to go too much into sort of a reference genome rant here right now, but it's just a standard way for us to look at a genome and say, okay, if we know that the reference genome for cats all say sort of ATCG in this region, and Lil Bub has AACG, then we know that that's an area that we should go look at further that might be a mutation. Reference genomes are also constantly being updated. So as sequencing technology gets better, as we learn more about what's happening sort of in our genomes and what is sort of that normal or standard, we're updating our reference genomes in these big repositories online so that we can keep all going back to it and checking. Oh, yes, students would be burning through many pencils if they were trying to take notes on this right now. But the lovely thing is I think that I have set this up so that you can rewatch it later. I might not have, but I hope that I have set this up so that it'll actually be watchable later. Okay, so mutations or variants are changes from sort of what we would consider the normal genome, although normal is a tricky word there, but so from the reference genome is how I'm going to put that for right now. So a number of people, including Lordship Mayhem, Vither, and Seth, sent in questions really asking about the specifics of Lil Bub's mutations. So there are a number of different ways that we can sort of look at genes and figure out what genes might be associated with what phenotypes. So for example, Knowing Better has informed me that I can make it public afterwards. Thank you. Okay, great. So yes, and now I know better. So one of the, what was I saying? Oh yeah, so we can look for mutations in genomes in lots of different ways. So if we have a really big population and we want to say, okay, this half of the population all has brown hair, and this half of the population all has blonde hair, we want to figure out what are the genes associated with brown versus blonde hair. We can do something called a GWAS, or genome-wide association study, where we can sort of separate those two people into groups and look for places where their DNA is different in a way that says that, okay, those locations in the DNA might be associated with this trait. We can also do things called candidate gene studies, where, you know, if we want to figure out, this is a completely made up mutation, but let's say somebody has pink hair, right? And they're the one person that we found who has pink hair. Well, we can say, all right, well, we know that there are these candidate genes that are involved in hair color already. We can go in and specifically look at those genes in this person to try and figure out sort of what, if there's a mutation in one of those genes that's causing this pink hair. So that's sort of a candidate gene study. If we have a big family, and some members of that family have a trait or phenotype, and other people in that family don't, we can do sort of a family study where we can look at the changes in DNA between those family members. And that's usually really nice because we can look, they have sort of a similar DNA background already because they're related and they're a family. And we can look for changes that way. But in Lil Bub, there wasn't actually any family data available. So Lil Bub was abandoned as a kitten, so the scientists weren't able to go look at her parents, and she was also spayed, so she didn't have any children of her own. So what they did is they did whole genome sequencing on Lil Bub. So what this means is that they took a blood sample from her and they sequenced the DNA from the white blood cells in her blood, and they looked at what they read. Now, I want to go into some of the specifics of DNA sequencing a little later, but for now, the idea is that they looked at the order of those A's, T's, C's, and G's in her genome, and then they compared them to the cat reference genome to try and figure out where she had variants that were not in that reference genome. So once they had done this and they had sort of aligned her genome to the reference genome, the first thing they did was look for DNA that might be associated with that polydactyly trait. So these are the additional digits that she had on her paws. Now, polydactyly is a phenotype or trait that shows up across mammals. So we already know some regions of the genome that are associated with this. So they specifically went in and looked at something called the ZRS enhancer of the sonic hedgehog gene. And let's break that down. So an enhancer is a region of DNA that can help to sort of turn on or off a gene. And the sonic hedgehog gene, and we will get to that name in a minute, is a gene involved in sort of patterning and development, and we'll dive into that a little bit more later. But they knew that in other mammals, this enhancer and mutations in this enhancer were associated with polydactyly. And in fact, when they did this, they found a heterozygous change, which means that they found it in only one of the two copies of her genome, of an A to a G in this region. So they found a single base pair change in one of the two copies of her genome. Remember, each copy is 2.7 billion bases long. And they found that that one single base pair change was likely causing this polydactyly. And that's crazy to me. This is one of the reasons why I love genetics, is that out of those 2.7 billion bases, although times two, so 5 billion bases, a single letter change had caused this visible phenotype. So this mutation has been shown in other mammals to cause ectopic expression of sonic hedgehog. So that means that during development, sonic hedgehog is being expressed in a place where it normally isn't. And so that place is sort of on the paw here. So she's growing an additional digit, likely because, this is a great question. So likely because of that mutation. So Felipe just asked, how do you know a mutation is associated with a trait just by observing and doing statistical analysis? So this is a really great question. So we use a lot of different evidence to try and associate this gene. So again, there were lots of other studies that have previously associated this mutation with polydactyly. So it was likely something, I didn't dive into all of those papers, but again, you can do family studies, you can look at these large groups and say, okay, this group of another mammal, probably cats actually, and we'll get back to why it's cats. This group of cats all have polydactyly and this group of cats don't. This group of cats all has this DNA mutation and this group of cats doesn't, and so it is associated. Now this is an important thing that I want to get across, is that we really want to talk about association. It's very hard to, we don't often, I don't want to dive into a whether or not we're proving anything in science discussion, but we are associating this trait with this genetic change. Now we can also go in some types of lab studies and actually manipulate, say, some model organism to try and give it that same mutation and see if it also has this phenotype. So we can do experimental things to try and track these down first, but we take together a lot of different types of evidence to say, okay, well, we have lab experiments and we have association studies and we have family studies, and we have all of this evidence that says that this trait is likely associated with this genotype. So that's sort of how we sort of integrate all of this evidence together. Now, one of the things that I want to point out is that this mutation is actually found in Hemingway cats. So there's a colony of cats that is descended from like Ernest Hemingway's cats, and they all, or many of them, have polydactyly, which has been associated with this mutation before. And there are lots of different breeds of cats that also have polydactyly and have different mutations. But I just think that's kind of crazy that there's this herd of Ernest Hemingway's cat descendants that all have polydactyly, and that is an amazing genetic resource for us. I think that's really cool. So they found this mutation that they thought was associated with the polydactyly phenotype. And before I dive into the second one, all right, a lot of good questions coming in, and I want to hit some of those, but I want to get to the second mutation that they looked at next. So they wanted to see if there was another mutation that might be creating these osteopetrosis phenotypes, where Lil Bub had these sort of really thick, dense bones. So what they did for this is they actually looked at the whole genome, and they wanted to see places where there might be variation that could be associated with this. Now, they identified a total of 6 million SNVs, so these are these single nucleotide variants, and small insertions and deletions, as well as 424 larger structural variants. And they did a lot to actually go through and figure out which of these might be associated. So on the left side of this figure here, you can see that they called, 6 million SNPs from their DNA, and then they found that 65,000 of them were in these regions of genes that might actually be expressed into proteins. They looked at 32,000 of those, which might actually be coding versus splice, and I don't want to get into all of the details of that now, but just to say that they started off with 6 million, then they went to 65,000, then 32,000, and then they narrowed them down to 12,000 candidates, and then finally they looked for genes associated in human bone disease. They narrowed it down to 12 candidate mutations, and then finally they manually filtered those down to 2 different variants that they thought might be associated with this phenotype. So this sounds crazy, but this is how a lot of this work is done when we're looking for rare variants in either patients or cats with rare disease, or really anything that has sort of these rare variants. Is a lot of filtering that goes into this. So we find so many things, because remember, 2.7 billion bases, you're going to find a lot of mutations. I mean, again, mutation sounds like a scary word that like, oh, if you have a mutation, something terrible is going to happen. But in humans, depending on the estimates, and there are a lot of different estimates of mutation rate per generation, each of us likely has somewhere, this is going to be sort of a squishy range, but somewhere between 40 and 100 new mutations that show up in us in each generation. But again, 40 new mutations spread across 6 billion base pairs. It's unlikely that any single one of those is going to be hugely deleterious. So we have to do these big sort of filtering. Oh, yes, thank you. Trying to get things happening on the screen mid thought flow. We have to do these crazy big amounts of filtering to actually find what we think are the candidate variants that are associated with disease. So they did all this filtering. They filtered it down to 2. One of them, they had pretty good evidence. So this is ZMPSTE24, and I'll point out how different that gene name is from Sonic Hedgehog, and I promise we will get back to Sonic Hedgehog. So this was a gene that can cause mandibular defects in both human and mice when mutated. And because Lil Bub has some jaw defects, they thought, okay, this might be a possible candidate. But there are multiple features typically associated with human and mouse ZMPSTE24 deficiency that are inconsistent with Lil Bub's phenotype. So this is another place where we're pulling in information from other areas of science, from human studies, from mouse studies, from experiments to say, this gene might be associated, but we have a lot of evidence that it doesn't quite fit that right phenotype, so maybe it's not the right one. So then the other candidate they found was TNFR-SF11A, which encodes for something called RANK, which is a transmembrane receptor. And again, we'll get there in a second. But this gene encodes for a protein that is on some bone cells that are involved with sort of the uptake and reabsorption of bone. So our bones are constantly being sort of rebuilt and reabsorbed over time. And so this gene was involved in that process. And so, again, I'm going to try and pull up a graphic here. And this was their graphic trying to look at this. So if you look at C here, which is bone reabsorption versus bone formation, on the left you have osteoclasts. These are sort of the bone reabsorption proteins and receptors, cells. And on the right you have osteoblasts. These are building bone. So they said, okay, if RANK is knocked out here, it's likely that these osteoclasts that would be reabsorbing the bone are not doing their function as well, but the osteoblasts are still working. And so you're getting this sort of overgrowth of bone, which over on the right, you can see Lil Bub had this sort of osteopetrosis phenotype where she had these really thick bones and this dwarfism. So I want to also point out here that up at the top, this is very small, so let's see if I can make this a little bigger for you. But somebody had asked to see what it looks like when we're actually looking at this data. So this is sort of a final view of the data in a program called the Integrative Genomics Viewer. And up at the bottom here, I don't think you can see my cursor, so I apologize. But down at the bottom of figure A, you can see that there's a string of A's, T, C's, and G's. That is the reference genome. And all of those red and blue lines are little pieces of Lil Bub's genome that have been aligned to that reference genome. And in that black box, you see this one base pair deletion. That was the mutation that they found in Lil Bub, that at this place, in both copies of her genome, she was missing this one single base pair. Now that was a lot of information, so let me back up. So the idea is that you have in your genome, you have sections of your genome, which are your genes, which encode for these proteins. And as I mentioned before, they're read in 3 base pair segments. So if you add or delete a base pair there, you're going to mess up what's called the reading frame. It's sort of how you read those 3 bases at a time. So this can cause the gene to have what's called a premature stop codon, which is just to say that like the reading frame gets messed up and the RNA that is encoding for the protein says stop way before the protein is supposed to stop. So you end up getting a truncated protein. It's shorter than the healthy version of the protein should be, and it, depending on how truncated it is, it may not be able to do its full function in the cells. Now, Lil Bub had a homozygous deletion that caused a frame shift in this gene. So homozygous means that it was in both copies of her genome, both the copy that she inherited from her mom and the copy she inherited from her dad. And it was a frame shift, and so it resulted in a shorter version of this gene. And because this was a transmembrane protein, so it's supposed to be on sort of the inside of the cell, in that cell membrane, and the outside of the cell, it was actually missing one of those domains. And I believe in that figure, I'm going to pull this back up here for a second just so that I can check. And so you can see that star there is where her mutation was. So it's missing the TRAF binding sites and that oligomerization domain towards the end. Not super important for our discussion today, but in case you were wondering. So yeah, so one of the things that I think is really interesting here is that it was a homozygous mutation. And so they don't know much about Lil Bub's parentage, but when they looked overall at Lil Bub's genome, there was a high degree of homozygosity, which means there were a lot of places in her genome that were identical, meaning she inherited identical copies from mom and dad. Now, there certainly are going to be lots of places in your genome that look like this, but her degree of homozygosity was so high that it was suggestive of sort of more of an inbred population. Now, especially in cats, this might be because there was sort of a small population of cats in that area that were breeding with each other, or it might be just that because it's a breed of cat, it might be that there's more homozygosity sort of in that overall breed versus sort of being outbred with different kinds of cats. So I just thought that was kind of interesting that they found this high degree of homozygosity within her. And yeah, so those were the two mutations that they found that they think are associated with her genome. Now, I want to circle back around and take a quick look. Okay, yes, cool questions coming in. Everybody's excited. Ooh, ASMR genetic discussion. I thought about this. I thought about like ASMR lab videos, but we'll get to that at another time. So I want to circle back to Sonic Hedgehog. Now, I noted that Sonic Hedgehog is the name of a gene that Lil Bub had a mutation associated with in this enhancer region. But why is this gene called Sonic Hedgehog? One of the reasons that Lil Bub, I'm getting all off track. I'm going to take a sip of tea because I'm getting a little sick and my throat hurts. I hope you all have tea with you as well. Okay. Back on track. Sonic Hedgehog. So Sonic Hedgehog is the name of a gene, and when you find crazy gene names in biology, they're typically from Drosophila biologists. And I say this with so much love because I was a Drosophila biologist in undergrad. I love my fruit flies and I miss them so much. But Drosophila biologists really like to give genes crazy names. So Sonic Hedgehog was a name given to a protein that functions as a chemical signal that is essential for embryonic development. So if you have a mutation in this gene, which is important for development of the brain and spinal cord, eyes, limbs, and many other parts of the body, the resulting fly has these sort of spiky denticles, which look a little bit like a hedgehog. So there were 3 of these sort of hedgehog genes discovered around the same time, each given slightly different names, but one of them, the gene, so Sonic Hedgehog, the gene was named as such by Robert Riddle, who was a postdoctoral fellow at the Tabin lab after he saw a Sonic comic book his daughter owned. So it is really named after Sonic the Hedgehog because it was one of these hedgehog genes and they needed to give it a hedgehog-based name. Now this is kind of fun and cool, but it's now sort of propagated because it was first found in Drosophila. This name is sort of propagated through other animals and mammals, and so you also have a Sonic Hedgehog gene. We humans have a gene called Sonic Hedgehog. And this is great in that it's sort of fun when you're learning about and stuff, but there are some sort of ethical thoughts about genes like this. I mean, I remember talking to someone in grad school who was saying that, you know, can you imagine having to be a genetic counselor trying to talk to a parent or to a patient saying, you know, oh, well, your child or you have a mutation in Sonic Hedgehog. Like, that's not great. Again, because you know, I think we have to be careful in how we're talking about these things and being sensitive to people and patients and stuff. So some of these gene names are sort of really fun, but I wonder, things get changed over time. I wonder if this is one that will stay just because it's got such staying power. It's one of the sort of first genes you learn about, I feel like, in development and embryonic development, and so everybody knows Sonic Hedgehog. But I think a lot of these other gene names that have sort of these funny names, sometimes we like to find less funny names for them because, you know, we're in a time where more people are getting their genome sequenced, and I think we want to be sensitive to that. But in flies, they can stay. I don't mind talking about a Sonic Hedgehog gene in a fly. But there are also genes. One of my favorites of this is a gene involved in the heart that's called HERG. And I remember, like, I've just talked about this gene a bunch in reading cardiac papers because that's some of what my research was in, or all of what my research was in. And I kept wondering, like, what is HERG? And I looked it up, and it's the human ether-a-go-go related gene. And as soon as I saw that, I was like, oh, this came from flies. And so I looked it up, and ether-a-go-go is a gene that was named, I think, back in the 60s by researchers somewhere here in Los Angeles because there's a bar called Whiskey-a-Go-Go, and go-go dancing was popular at the time, and these flies who have a mutation in this gene, when you put them under ether, like, their legs danced and it looked like they were go-go dancing. And this is a crazy thing that is now propagated into a gene that's we refer to as HERG, and I understand why we refer to it as HERG, or some people say like HERG or whatever, but we don't talk about the human ether-a-go-go related gene because that's just a weird thing that has come from fly biology into humans. So that's a long, circuitous way to talk about Sonic Hedgehog. Somebody actually asked, I'm trying to figure out who it was, Electromorphous asked, do they have to ask for the copyright for using game character names for naming mutations? I tried to look that up, and I actually couldn't find anything on it. I don't know. I think they just did it. I don't think anybody asked. I think the fly biologists, and again, I love fly biologists. I miss being a fly biologist. I think they just went for it. I think they just did it. I don't think they actually asked any kind of permission. So, all right, I'm going to take a quick... Yeah, so crazily named genes probably are more memorable, and I agree that it's a lot easier to talk about something called Sonic Hedgehog than it is to talk about something called TNFRS F11A. So I think that there has to be a good middle ground, right? That you have to find something that's easy to talk about and easy to communicate about, but maybe isn't associated with a video game character. So I think that there's a middle ground here that we should sort of be striving for. It's not really one or the other. Oh! Somebody just let me know I just passed 5,000 Twitter followers. Very exciting. Thank you all. That's insane to me. Absolutely insane. All right. Oh yeah, isn't there an issue regarding trademark of Sonic Hedgehog? If somebody knows, please let me know, because I really did try and look this up and I couldn't find anything that said that they had to ask for permission or anything. But yeah, I'm curious to know. Okay, so we're already over my thought that this was only going to be 20 minutes long. I'm sorry, I'm just babbling about genetics, but again, this is one of the most fun things to me. So the last thing that I think I'll touch on here is how you sequence a genome, and this is a question that was sent in by James, who really wanted to know sort of how we do this and what the data looks like. There are a number of different ways to sequence genomes now available to us, but the most common overview is that you take a sample from somebody or something. So in this case, they took a blood sample from Lil Bub. Then they extracted the DNA from that sample, which really sounds crazy, but really you're just breaking open the cells and you're purifying the DNA sort of out of what comes out of those cells. You take that genome and then you make a lot of copies of it and you break them into little pieces. And then you go through each little piece, you put it into a machine called a sequencer, and they do these in different ways, but you read base by base by base what is in that little piece of DNA. And most of the popular ones do this using light. So you have your two pieces, your two strands of DNA that you've separated apart, and you rebuild the complementary strand. This is getting a little bit into DNA biology, but you rebuild the complementary strand, and each time it puts out a different light signal based on whether it's adding an A, T, C, or G. This is a huge oversimplification, but you read base by base what's in those little chunks. So now you have all of these little fragments, and again, Lil Bub's genome, one copy was just 2.7 billion base pairs long, and you're reading it in something that's about 150 to 250 base pairs at a time. So you're really taking these little chunks. And then you have your reference genome, if you're lucky. Some species don't have a reference genome. And then you align all of these little pieces to it, and you reconstruct Lil Bub's genome of billions of bases from these couple hundred base pair chunks. So it is like taking a billion letter long book, having 40 copies of it, chopping it up into 150 character pieces, and then putting them into a giant pile and trying to rebuild the book. It's kind of wild that we can do this. And there are new techniques that do longer chunks, and lots of pros and cons to each of those. So I actually, in my PhD, typically worked with the longer types of sequencing, so I have a special fondness for them. And there are lots of different companies who do this and some different specifics to it, but that's really what you're doing, is you have a sample, you break it up into lots of pieces, you read those little pieces, and then you put it all back together. And now what you can do is you can look letter by letter by letter by letter at what's in that genome, which is pretty cool. I think it's pretty cool. I'm looking at if there are any last questions. Oh yeah, reads and contigs my daily life. Yes. So if you're someone who does this, you know each of those little pieces is called a read. Oh, and I meant to talk about what the data looks like. There are many different file types, but at its core, what you have are really essentially text files that have those 150 to 250 base pair A, T, C, and G chunks. That's what it looks like. Depending on what sequencer you have and exactly what file type it is, there's often some information about the quality, maybe some information about some other stuff, but you get back lots and lots of text files of A's, T's, C's, and G's. And it's cool. It's really cool that we can do so much with those, and it's really powerful. And I mean, people talk a lot about the fact that when we first sequenced the human genome, it was, $1 billion and took 10 years and stuff. And now you can have your genome sequenced for under $1,000 in a couple weeks, and that's really just processing time. I mean, you can do it in like a day or two, really. It's gotten very cheap, and it's really easy, pretty easy to do, and you can get a lot of information from it. But yeah, so genome sequencing is cool, is great, and in a situation like this, so again, Lil Bub was a cat, but I think there are a lot of parallels here from Lil Bub's genome sequencing to the sequencing that we do in patients, which is, again, really something that I'm passionate about, is sort of better sort of sequencing of patients in precision medicine, and how do we figure out if you have a rare mutation associated with the disease. And so, yeah, if we don't have family history like we didn't have for Lil Bub, and we don't have sort of information about siblings and children and stuff, doing something like this, doing whole genome sequencing, and then using all of the information available to us in the literature, from experiments, from association studies, to try and pull all of that together to figure out what are the mutations that are associated with the phenotypes that you have or that Lil Bub has. I think that's super powerful, and I was so excited to be a geneticist, and am a geneticist, at a time where this is a thing that we can do, because it's so powerful, and I think just really exciting to be able to take that deep of a look at your genome. So, final look at the astronaut on your shelf is, the astronaut on my shelf, that's actually a postcard from a bar in San Francisco that had arcade games that I don't remember the name of, but they were giving out cool postcards when I went there like 2 years ago, so I grabbed that postcard. Other questions? Oh yeah, scientists are bad at naming things. The blue-footed booby bird is an example. Sure. I mean, names are important, right? How you name things, how you discuss things are very important. However, I would argue that the blue-footed booby is a very popular bird, and would be popular because of its fantastic blue feet, but is maybe more popular because of its name. And I'm going to count the BlueFooted Booby as the reason why this stream is not for children. So you know, YouTube. The Pokemon gene, the researchers were threatened with a trademark infringement suit by Pokemon USA. I will have to look into that. I'm sure they were. That's crazy. A Hugo committee. Oh yes, somebody. So there is a somebody. Mike Downs is looking into the Hugo committee procedure on the naming, which is great. I looked at that really briefly, but it is one of the genes that's brought up as something that we should probably think about finding a new name for, to be able to discuss it ethically with patients and not feel weird about that. But yeah, I have gone way over the 20 minutes I thought I would do, but I think this was fun. I don't know if you guys thought this was fun. You guys sent in so many really cool questions. I want to do this again. One thing I will point out, though, is that currently this is called science news, because I am terrible at naming things. This is not one of my strengths. So if you have any better suggestions for what to call this two-part series, I think this was a success. I think this was fun. I want to do this again and talk with you guys. And so it just needs a better name. It can't be science news. That's too on the nose. So please, if you have any suggestions for names, please let me know. This video will be archived on the channel. I will figure this out. Oh, Sonic the Hedgehog science stream. I kind of like that. Yeah, I want to do these again. Please leave me names. If you have a paper that you would love to see this done on, send me those suggestions as well. I don't think this is going to be a weekly thing, but I'd love to do it once or twice a month. I think would be really cool. And as a reminder, there are a lot of great resources down in the description of this video. Both the really kind videos sent in by the researchers who are part of this study, who have made their own videos about this, and also the original papers and all sorts of stuff. If you do want to sort of keep diving into this, there's a lot of great resources down there. And Alex updates. I don't want my name to be part of it. I feel like I want this to be about the science, not about me. But something with updates or like discussion, because I think like that's the part of this that I really want to, now I'm just babbling, but that's the part of this that I really want to get out of this, is actually talking with you guys about science, not just at you. So something about that discussion. I'm throwing that out there for you. But yeah, this was super fun. Thank you guys for joining me so much. I really appreciate that you hung out with me for 47 minutes today to talk about genetics. This was great, and go forth, do science. I'll see you again soon. Happy Valentine's Day. Have a great weekend. And yeah, go forth, do science, and talk about cats. All right. Bye, guys.